Hi guys, today we're solving Cambridge A-Level Biology Paper 1 MCQ Topicals for Cell Membrane and Transport, that is Chapter 4. Question number 1. Which statement describes carrier protein in cell surface membrane? So the two transport proteins we know are carrier proteins and channel proteins. Let's see what we know about carrier proteins. Carrier proteins they bind to specific molecules. There will be a conformational change. They transport larger polar molecules. And some examples would be glucose, amino acids. So there's specific carrier proteins for glucose and specific ones for amino acid, each one. And this can either be passive in facilitated diffusion or it can be active, like in active transport where it requires energy, ATP. Whereas channel proteins are going to be described as water-filled pores that are hydrophilic. They're going to look like an open tunnel. They transport small charged particles like ions, and it will be down the concentration gradient. Now let's look at our options. Carrier proteins are found on the outer surface of the cell membrane, allowing cell recognition. So that's not carrier proteins. We know that's lipids, glycoproteins. Carrier proteins are involved in moving substances through the membrane by either active transport or passive transport. This is true. Carrier proteins allow the binding of ligand molecules, which causes changes within the cell. Again, this binding, cell signaling, all of that is for receptor proteins or glycoproteins, glycolipids. The last option, let's see why this one is wrong. Carrier proteins are involved in moving substances through the membrane by passive transport through water-filled pores. So that water-filled pores, anytime this wording is used, you think of channel proteins. Question number 2. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential than the water potential inside the cell? So what's that going to look like? The cell outside, the water potential is higher, and inside the water potential is lower. When you talk about the solute, we're going to say that it is going to be higher inside because water potential is lower, and lower on the outside. So you should be able to figure out all of this. Now let's look at our options. The bacterium bursts because the cell wall has no structural function. That is wrong because its cell wall is made up of peptidoglycan. So this would be wrong. The bacterium dies because water leaves the cell by osmosis. The movement of water is going to be inside, not outside. So water is not leaving the cell. There is no change because the cell wall is impermeable to water. This is wrong because cell wall is fully permeable to water. D is our answer. There is a net movement of water into the bacterium. This is what we see here. Question number 3 is testing your theory from chapter 9, I think. By which process do hydrogen carbonate ions leave red blood cell? So hydrogen carbonate ions, when did we learn that? So this was in the process of chloride shift. What happens is inside the red blood cell, so you've got a red blood cell, carbon dioxide moves in, and there is an enzyme, carbonic anhydrase. Carbon dioxide reacts with water and forms carbonic acid. This is carbonic acid. Then this carbonic acid dissociates to form hydrogen carbonate ions and hydrogen ions. And then from this red blood cell, the hydrogen carbonate ions will move out and chloride ions would move in. So this process of the hydrogen carbonate moving out, this we know is this is a passive process. So it's passive, and it's gonna be down the concentration gradient. What else do we know? We know that this uses a transport protein. So the first option is active transport. This is wrong because this is a passive process, not active. We don't need ATP. We're not going against the concentration gradient. This is not our option. B is endocytosis. This is wrong because endocytosis is like movement into the cell when membrane engulfs large particles to bring them into cell using vesicles. So that's not what's happening here. C would be our answer. This is facilitated diffusion because it's using transport proteins. This can either be carrier proteins or channel proteins. The last one is not our answer because again, it's a type of endocytosis where the cell engulfs solid particles, just like in the case of white blood cells engulfing bacteria. Next question. In an experiment, fluorescent dyes were attached to proteins on the outer surface of cell surface membranes. Fluorescent dyes of one color were attached to proteins of a living human cell, and fluorescent dyes of a different color were attached to proteins of a living mouse cell. The human cell and the mouse cell were then fused to form a hybrid cell. So we learned about hybridoma cells. Similar thing here, but they are not going to be testing you on that. This is more about the cell surface membrane. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. So this is the key information. It's distributed randomly across the hybrid cell surface membrane. What does that tell you about? What does this experiment show you about the cell surface membrane? The fact that proteins are able to move freely, that's how they were able to be randomly distributed. So it's this option. C. Proteins move freely in the phospholipids of a bilayer. And why is B wrong? Because proteins in the outer layer of a bilayer do not penetrate into the inner layer, so that's wrong. And the last one, it's true because the cell surface membranes of two cells are bilayers, but this is just an irrelevant fact. So a lot of the time you will get irrelevant facts to confuse you, but you should read through all of your options and then decide. This question is a little tricky. A maize crop is growing successfully in a field in which the water potential of the soil is -40. What is the most likely water potential of the cell sap in the root hair cell? So the root hair cell should be less. So it should be lower water potential. Should have lower water potential in the root hair cell. Only then the water will move into the root hair cell from the soil. Because here they have said it's growing successfully. So for that, it's not going to be dehydrated. So normally it would move from the soil into the root hair cell. For this, it needs to be low water potential and the soil has to be higher water potential. So straight up, we can get rid of this option because it's the same as the soil. If it's equal, then there would be no net movement. And we can get rid of this and this because, so when they talk about negatives and water potential, it gets a little confusing, but you can remember it like for osmosis, higher water potential is going to be the one that is less negative. And lower water potential is going to be the one that is more negative. So this should help you do questions like this that come up a lot frequently. So over here, both 0 and -20 is less than the one we have for soil, less than -40. So because it's less negative, then it's higher water potential. So in this case, it's higher than soils. So if we picked either of these options, the water would move out of the root into the soil. So this is our answer. Why? Because -60, this is higher. So it's cell sap moves. Cell sap is more negative than -40. So if it's more negative, that means it's lower water potential. So it has to move from lower water potential to higher water potential to lower water potential. So this is our answer. So the next page. Question number 6. Which statement suggests that a membrane protein is involved in active transport? So membrane protein in relation to active transport. So active transport, you know that it's against concentration gradient. And it uses ATP. So let's look at our options. It allows movement of molecules across a membrane if concentration difference exists. So this is just wrong because it's describing more of passive, which is just simple diffusion. When there's a concentration gradient, then it's just moving from high to low down the concentration gradient. B, it can only function if mitochondria are supplied with sufficient oxygen. So in this case, oxygen is required. If there is less oxygen, because oxygen is needed for aerobic respiration, and if there is less aerobic respiration, then mitochondria is producing less ATP. So it can only function if mitochondria are supplied with enough oxygen. This is our answer. C, it has a tertiary structure with a binding site with a specific shape. So that is irrelevant again, because although this is like true for carrier protein, it's specific, similar and enzymes. So, but this is not related to active transport. So our question was active transport. We're gonna get rid of this option. D, that's not the answer. It is found in the cell surface membrane and the mitochondrial membrane. Again, it is found, the membrane protein is found, but that's not telling us anything about the active transport. So B remains our answer. Cell surface receptors used in cell signaling are usually proteins that extend from one side of the protein to the other side of the protein. What is the role of extracellular part of the protein? So this is specifically asking about the extracellular part of the protein. And this is important. So the extracellular part alone, that is for binding the cell signaling molecule, because it has a specific shape that ligands bind to. Ligands will bind to. Ligands are the cell signaling molecules. Functioning as an enzyme, this is wrong because we're specifically talking about the extracellular part. So this would be wrong. Increasing the solubility in the receptor, that is, although it has to be hydrophobic, part hydrophobic to fit within the phospholipid bilayer. That is a structural property, so this is not our answer. Transmitting the signal from the target cell, it does transmit the signal, but not from the target cell. It transmits into the target cell. Question number 8, which substance can pass directly through cell surface membrane and do not use carrier proteins or channel proteins? Pass directly through cell surface membranes and do not use any of our transport proteins. So let's look at our options. These are both ions. What are ions? They are charged particles, and they would repel. The hydrophobic core of the phospholipid bilayer would repel them, so they need to be transported through channel proteins. So in question number 1, we discussed what's transported through carrier and channel proteins. Ions typically are transported through channel protein. So this is not going to be directly transported. Carbon dioxide, yes, because it's small and it's non-polar molecule, so it will directly transport. This is glucose, so hopefully you can identify that. C6H12O6, that's glucose, and glucose is large polar molecule, and we learned earlier that carrier proteins, there will be specific carrier proteins to transport glucose. So glucose is transported through carrier protein. It's a large polar molecule. So the only one here is 2. That's D. A student put a layer of plant epidermal cells on a microscope slide. The student put a drop of potassium nitrate solution on the layer of cells and observed that the cell surface membrane of many of the cells had separated from the cell wall. So they've separated from the cell wall. This is a plant cell we're talking about. And the cytoplasm and cell contents had shrunk. So cytoplasm and cell contents had shrunk. The cell surface membrane had separated from the cell wall. And this is a plant epidermal cell. So what are they asking? What explains these observations? Let's draw a diagram to make it easy. So if you draw something like this, you have this cell, and inside the cell the water potential is going to be high and outside is going to be low for it to move outside. And when the water moves outside, then these changes would be observed. So where is the water moving? The direction of net movement of water is going to be from the cell to the solution, not outside to inside, it's inside to outside. So it's either this or this. And the water potential of cells at start and water potential of solution at start. So looking back at this note that we wrote down earlier, if it's higher water potential, it has to be less negative. So inside the cell, it's higher water potential. Water potential of cell, it's going to be higher water potential, that's less negative. So it's going to be -100. And the other one is going to be -500 because this is lower water potential, so more negative. Lower water potential, more negative. So A is our answer. Question number 10. High concentration of ethanol can disrupt cell surface membrane structure and make the cell surface membrane leaky. Yeast cells release ethanol as a waste product of metabolism. Yeast cells can alter the composition of the cell surface membrane to reduce the effect of ethanol. So they're going to alter it so that the ethanol does not make it leaky. So what changes are made? A saturated fatty acid X is placed by an unsaturated fatty acid Y. Y has shorter. So saturated means it's carbon-carbon single bond, and unsaturated is carbon-carbon double bond, and it's going to have kinks. So Y has a shorter hydrocarbon chain than X. There is an increase in phospholipid to protein ratio, so there's more phospholipid than proteins. Which statement would explain why the effects of ethanol is reduced? So how do these changes reduce the effect of ethanol? By making it less leaky and maintaining the stability, basically. So a greater proportion of phospholipid may increase the entry of ethanol via channel protein. So it does not increase the entry, that would be wrong. B, an increase in hydrophobic interaction by phospholipid tails improves the membrane stability. This is correct because what's happening here is it alters altering the composition of the cell surface membrane to reduce these effects of ethanol, and it makes it more leaky. So in this case, we do the first change, we replace an unsaturated fatty acid with saturated, then we get rid of the kinks, so it's packed more closely together. More close together. An increase in hydrophobic region by phospholipid tails improves the membrane stability. So that's our answer. Why not the other one? Because an increase in the proportion of fatty acid chains with double bonds. So there is not increase of double bonds. We got rid of the double bond unsaturated fatty acid, replaced them with saturated single bond ones. And then D, the shorter hydrocarbon chains of Y decrease membrane fluidity. They don't decrease membrane fluidity, they increase it. Next question. The photomicrograph shows the appearance of onion epidermal cells, plant cell, after they have been soaked in solution X for 1 hour. What fills the space labeled Y? So this space, what would fill this space? First of all, this is onion epidermal cell, so that tells you immediately you have to think of plant cell. And what this looks like is it's plasmolized. Going over the theory, this is what happens. So this is a turgid cell, and then when you have water moving out, then the cell looks something like this. It's plasmolized. It starts to plasmolize, and then when you have it like this, it's fully plasmolized. So what happens here is protoplast has shrunk away from the cell wall. The cell is fully plasmolysed. What would fill these spaces? These spaces are not empty, it's not filled with air. The solution will actually fill these spaces. So the answer here would be solution X, not air. Don't make that mistake. Equal sized potato pieces were placed into a test tube and covered with a sucrose solution. The test tubes was left for 30 minutes. All other variables were controlled. After 30 minutes, the potato pieces had not changed in size. So there is no change. What can be concluded from this results? Equal sized potato pieces left for 30 minutes. Everything else was controlled in a sucrose solution. And then they had not changed in size. So what can you conclude from this results? Let's look at our options. The concentration of sucrose is the same. That would be wrong because we're not talking about the concentration. It's likely to be the water potential. So the water potential is the same in the potato and in the source sucrose solution, and there is no, so the first bit is correct. But you can see both in this case and the other one, it's the same first part. The water potential is the same in the potato and in the sucrose. But carefully read the second part. The second part for C is there is no more movement of water into or out of the potato cell. So that wording is wrong. It's supposed to be there is no movement, not no more. So it's no net movement of water into or out of potato cell. So this is our answer. The diagram represents part of the fluid mosaic model of a cell surface membrane. They've given you this diagram. Which part of the membrane are hydrophilic? Hydrophilic parts from this fluid mosaic model of cell surface membrane. So let's label them first. You have one, this is your glycoprotein. That's hydrophilic. Hydrophilic. You have your 2 is the protein. And this is hydrophobic because it's embedded into the phospholipid bilayer. 3 looks like the phosphate head. We know this is the phosphate head. Phosphate head. And 4 is the fatty acid chain. Phosphate head is hydrophilic, and this is hydrophobic, forming the hydrophobic core of the phospholipid bilayer. I'm sorry, I don't know why I wrote hydrophobic here. This is hydrophilic. So again, that's a common mistake. You might think this is hydrophobic, but it's facing outwards. So it's supposed to be hydrophilic, this bit. So you have 1, 2, and 3 as your answer. That's A. Which statement about the proteins and glycoproteins in cell surface membrane are correct? So proteins and glycoproteins in cell surface membrane are correct. They can allow cells to bond together to form tissues. Yes, because remember that glycoprotein will act as addition molecules. The second one, they can recognize messenger molecules like hormones. You know that because membrane proteins and glycoproteins, they will act as receptors with specific shapes, and they act as cell so that they can specific shape. So they can recognize messenger molecules like hormones, the cell signaling. They can be antigens and allow cell-to-cell recognition. Yep, because they will allow the immune system and other cells to recognize cell versus non-self cells. So all three would be correct. So it's A. Which pair of factors is inversely proportional to the rate of diffusion? So what we know about rate of diffusion is that it's a pair of factor. There are two factors, right? Inversely proportional means that one is increasing, the other is decreasing. So what we know about this is the rate of diffusion is inversely proportional to the distance over which the diffusion occurs, and also inversely proportional to the size of the diffusing molecule. So let's look for that. Distance over which molecules occur and the size of diffusing molecules. So that's B. B is our answer. 16. Raisins are dried fruits that contain high concentration of sugar. Which row is correct when the raisins are first put in the water? What's going to happen to the water potential in the raisins compared to the surrounding water and the direction of movement? So let's draw a diagram for this. If you have a cell, we have raisin instead of cell. You have low water potential in the raisin because you have high solute present. There is high concentration of sugar, and outside there is low concentration of sugar but high water potential. The water movement is from out to in. So let's get that straight. The water movement is going to be into the raisin. These two options, and the water potential in the raisin compared to the surrounding water has to be, so referring back to this note, if there is low water potential inside the raisin, then it's going to be more negative. So this is our answer. Lower water potential. The diagram shows the movement of substance Z across a cell surface membrane. This is inside and this is outside. So the movement is, you can see over time, it's inside the cell and then it's here and then it's outside the cell. So the movement is going outwards. This is moving out. Moving out is exocytosis. So just looking at this, what we can tell is this is a vesicle that fuses with the cell surface membrane, and then and then it's sent out, it buds off. On the other end, this is bulk transport out of cell, that is exocytosis. So endocytosis is wrong because endocytosis is into cell. Exocytosis is correct. And these two would be wrong because there are again both types of endocytosis, which is movement into the cell. Whisking tubing is often used as a model during experiments to investigate osmosis in plants. What could whisking tube be used to represent? So remember, whisking tubing or dialysis tubing could be used in experiments to represent a partially permeable membrane. This could be the cell surface membrane or the tonoplast, because tonoplast is also a cell surface membrane for the vacuole. It's a membrane around the vacuole. So it's tonoplast or cell surface membrane, but not cell wall, because cell wall is fully permeable, so that's irrelevant. It's going to be B. Question number 19. The fatty acids Lactic acid and oleic acid have exactly the same structural formula with one double bond in the chain. You can see the double bonds here. The shapes of the chain are different as shown in the diagram. So this is straight and this is this way. Which row shows the effect of presence of these fatty acids on the structure and behavior of cell surface membrane? On the structure and behavior of the cell surface membrane, the effect of these fatty acids. So they have the same structural formula with one double bond in the chain, but the shapes are different. So what options do we have here? It does not fit closely with other fatty acids. So this one does not fit closely together. That would be wrong because it will fit closely together. This is just a straight chain. So this is wrong. It fits closely. This one would not fit closely together because it's not a straight chain. It has kinks. So this would be not our option. B is does not fit closely together. Again, this is wrong and this is wrong. Also, they are talking about fluidity. So when it does not fit closely together, it won't be less fluid at high temperatures. So that's also wrong. C is fits closely with other fatty acids. Fits closely with other fatty acids so that the membrane is less fluid at high temperatures. So this is correct because if it fits closely together, it's packed closely together, and that is going to be when temperature is high, it's going to make sure that it doesn't get too liquidy, it's less fluid. So this is correct. And then for the other one, it does not fit closely together. That we know because this has kink. So with other fatty acids so that the membrane is more fluid at low temperature. So this is also correct because then it prevents it from freezing. We don't want that at low temperatures. So it's going to keep it more fluid. This is the answer. The diagram shows a simple cell signaling pathway in which a cell signal molecule leads to a response, such as secretion. So you have cell signal molecule and then you have a response, and what's happening in the middle, that's going to be, you have activated enzyme in cytoplasm. That's not happening immediately, so this is wrong. Target in cell surface membrane is wrong. Lipid in cell surface membrane, signal molecule, there will be a receptor protein, and this can either be lipid or protein in cell surface membrane. So it will attach to that. And then what's going to happen next is not an extracellular enzyme, but an activated enzyme in cytoplasm. So it will activate, it will enter the target cell and activate the enzyme in the cytoplasm of the cell. So it won't target in cytoplasm, that's wrong. Target cell is what we talk about. So C is our answer. This was about cell signaling pathway. Okay, let's label these structures. It shows a cell surface membrane. So cell surface membrane, you can see this is a phospholipid bilayer and another one, so it forms a phospholipid bilayer, that is cell surface membrane. So what is Y? Y is the glycoprotein or glycolipid. This is for cell signaling and cell to cell recognition. You have X. Remember in the first question we noted that the channel protein is going to have this tunnel sort of shape, and it's going to be water-filled pores. So that's this is channel protein. X is channel protein. W is phosphate head, that the hydrophilic phosphate head. You have this thing in the middle, that's Z. Z is not, you see these two are phospholipids. This is also a phospholipid, but the other one is not phospholipid, doesn't have the phosphate head. Z is cholesterol. That's everything here, which is a correct role for a labeled molecule. W is involved in controlling membrane stability. W is not for membrane stability, that is cholesterol. That is Z. X is involved in active transport. X is channel protein. So channel protein is not involved in active transport. It is carrier protein that is involved. Y is involved in cell signaling. That is our answer. Z is involved in diffusion of ions. That is wrong because Z is a cholesterol. It has nothing to do with transport. The cell surface membrane, this is the one. We've done this. The diagram represents the fluid mosaic model of membrane structure. Again, similar question. So let's look at what we've got here. 3 again is glycoprotein. You have cholesterol again here as 4, and 2 and 5 seem to be the proteins. They look like extrinsic or peripheral proteins because they're not spanning throughout the cell surface membrane, the bilayers. It's just present on the outer surface like this. 6 is the fatty acid chains, and 7 is the phosphate head. What are they asking? Which 2 of the numbered components contribute to the fluidity of the membrane? So when we talk about fluidity of the membrane, it has to be which 2? So fluidity is going to be, it's cholesterol, this, and it's going to be the fatty acid chains, because fatty acid chains, whether they are saturated or unsaturated, that's going to contribute to this. The cell surface membrane of some cells are largely made up of phospholipids and cholesterol, with few proteins. Which transport mechanism will be reduced across this membrane? So which one will be reduced? Transport mechanism. Facilitated diffusion will be reduced because facilitated diffusion is through carrier and channel proteins. So because there are less proteins, you're going to be reducing facilitated diffusion. Active transport again is going to be reduced because this is through carrier proteins. Diffusion does not require, simple diffusion does not require proteins, so it's not going to be affected. So it's 1 and 2B.